CBSE, Vedantu, NCERT solutions, and in this channel we do NCERT line by line. We make solutions and we find solutions to each and every question given your NCERT also. Now students, this is the part 2 of the cell unit of life. In the part 1 we started off with the basic introduction about a cell, that is the overview of the cell. Then we also did something called as prokaryotic cell, we did eukaryotic cell. Under eukaryotic cell we started off with plasma membrane, cell wall, endoplasmic reticulum. So until then, it is all covered in the first part, done and dusted, every single line of NCERT, right? Now in part 2, what are we going to do in part 2? In part 2, the remaining of chapter, we'll be starting off with Golgi apparatus, where we have to study, students, this extreme part here. Can you see? This is the part where you need to focus. That is the entire structure of Golgi apparatus. This we'll be doing in today's class, the entire of it. The entire chapter will be over in today's class. So with that being said, students, welcome to NCERT solutions by Vedantu. Right. Now let's read what is about Golgi apparatus first. Let's read the NCERT. Then I will explain you what exactly is the function of Golgi apparatus. Okay. Golgi apparatus. Okay. Let's start, students. Hello, 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 and happy Ganesh Chaturthi to everyone. Okay. Golgi apparatus. Camillo Golgi in the 1898 first observed densely stained reticulate structure near nucleus. That is, if this is the nucleus, friends, if this is the nucleus if that is the nucleus right there, 1 second. Nucleus has how many layers? Can anyone tell me? Nucleus has 2 layers, outer layer and inner layer, right? Outer layer and inner layer. So this is the outer layer. And all of you know, in the previous class I told you, from the outer layer, there is always endoplasmic reticulum is coming from the outer layer. Now near the nucleus itself, you can find the Golgi bodies. Golgi bodies can be found near the nucleus itself, right? Near the nucleus. Okay. So that is Golgi bodies. They were later named Golgi body. Why? Because the first observer was Golgi, Camillo Golgi. It's a PYQ. So if they ask you a question, who discovered the Golgi apparatus? You're supposed to say Camillo Golgi, right? Camillo Golgi discovered the Golgi apparatus. Simple question, direct question they'll ask you. Okay. They consist of many flat disc-shaped sac or cisternae. Right? Cisternae is present. That is disc-shaped long stacks which are arranged parallel to each other. Right. These are stacked parallel to each other. Varied number of cisternae are present in Golgi complex. That is, number can vary. Right. This long structure, cisternae, the number can vary. Right. The number can vary. We can see structure. This is the cisternae. The number can vary. Okay. Now the Golgi cisternae are concentrically arranged near the nucleus with a distinct convex and convex cis or the forming face. We have the cis face and the concave trans face. Understand, students? Now here, if you look at this is the Golgi body. This is how the Golgi body looks like. If you look here, this part is called as the cis face. This part is called as cis face. Cis face is also your convex face. Can you see here? Convex face. Convex face. Also called as the forming face. Also called as the forming face. Understand very well. It's very important concept here. This is called as the trans face. Trans face. Trans face is also called as concave face. Also called as maturing phase. Also called as maturing phase. Now understand how they are arranged with respect to the nucleus. Understand? Okay. This right here is the nucleus. This right here is the nucleus. Outer layer and inner. 2 layer. Outer wall, inner wall. Okay. Now on this outer wall, we have the endoplasmic reticulum. Endoplasmic reticulum is there. Now from this endoplasmic reticulum, here we'll find the Golgi bodies arranged concentrically. Golgi body. This is the Golgi body. This is the ER. This is the nucleus. This is the nucleus. Okay. So all of you are aware that a nucleus and we have endoplasmic reticulum. All of you know from endoplasmic reticulum we can get 2 things. If it is RER, if it is rough endoplasmic reticulum, it can produce proteins. Now what about smooth ER? Smooth ER can produce lipids. Here we learned here, right? See here. Rough ER produces proteins. Rough ER can produce the proteins, while smooth ER produces lipids. Now, whatever lipids and proteins which is produced, for example, here we have proteins and lipids are coming out from here, from the ER. The main function of the Golgi complex or Golgi body is to completely package it and send it next step. For example, your Golgi body is like Amazon factory. It is like Amazon factory. Endoplasmic reticulum is producing the proteins. Golgi body will add a cover on top of it, completely nicely package it, and it will give it to the next one wherever it's required. It will send it to wherever it's required. So for example, if Golgi body is making the proteins, this is the proteins, right? Proteins, the protein will be first entering. It will be first entering this phase right here, where the entering phase is called as the cis phase. Is the cis phase entering. So can you see here in this diagram also, if you notice, in the cis phase we have the incoming transport vesicles. Incoming transport vesicles from the endoplasmic reticulum. So products of endoplasmic reticulum is first received by the cis phase. Received cis phase. Then it gets completely packaging. Packaging takes place. Once it is completely packaging is done, it exiting is by trans phase. So this outside part here, this is called as the trans phase. Trans phase. Exiting is called as the trans phase. Entering is called as the cis phase. What is the main function of Golgi body? Packaging. For example, your ribosomes, rough ER is making protein. To this protein, students, to this, if your RER is making some proteins, right? It is making some proteins. To these proteins, your Golgi body will add a carbohydrate and send it out. Let me show you the example here. Okay, let me show you the example here. Okay. See here, first of all, just read what we learned here. See, concentrically arranged near the nucleus and distinct convex or forming phase. See, convex or the forming phase or the cis phase. Other one is called as what? Maturing phase. That is concave is the concave or trans or maturing phase is the where it is coming out. Okay. The cis and trans phase of organelles are entirely different but interconnected. Very PYQ line again, PYQ line. The two phases are different but they are interconnected. Okay. Now, the Golgi complex principally performs the function of packaging. I told you, Amazon factory it is. Okay. To be delivered to either to the intracellular targets, that is within the cell, or secrete outside the cell. Materials to be packed in the form of vesicles from ER. See, product is coming from the ER. From the ER with the cis phase of the Golgi apparatus and move towards the maturing phase. So they are entering where? Entering in the cis phase, nicely packaging is done, removed from the trans phase. This explains why Golgi apparatus remains in close association with the endoplasmic reticulum. That's why wherever you find endoplasmic reticulum, near endoplasmic reticulum, there will be Golgi complex. Okay, that point is clear to all of you? So wherever you find endoplasmic reticulum, Golgi complex will be along with it. Okay. A number of proteins are synthesized by ribosomes. I told you. RER is producing the proteins. To these proteins, your Golgi body is adding a carbohydrate like glyco and will end up becoming glycoprotein. For example, smooth ER, smooth endoplasmic reticulum. Smooth endoplasmic reticulum is producing lipids. To these lipids, Golgi complex is adding a glyco part. That is carbohydrate part, glyco part. So what do we get? We get glycolipids. So glycolipids and glycoproteins are the products of your Golgi complex. Why? They're packaging everything. Okay, they're packaging everything. See here. On the endoplasmic reticulum are modified in cristae of Golgi apparatus before they are released in the form of trans phase. Golgi apparatus is the important cell for formation of glycoprotein and glycolipids. Glycolipids are made by your Golgi complex. Protein is from your ER, lipids from ER. but packaging the glycopad is added in the Golgi body. In the Golgi body. Okay, very simple. Golgi body. Now, lysosomes. Lysosomes. First of all, let me tell you, lysosomes are a product. They are a product of Golgi complex. Basically, packaging material. No, lysosome is one of the products of your Golgi complex. Okay, what is Golgi complex? Understand. These are membrane-bound vesicular structures. So they are in the form of vesicle, membrane-bound, formed by the process of packaging in Golgi apparatus. I told you, Golgi apparatus is packaging. It is giving it here. Okay. The isolated lysosomal vesicles have been found to be very rich in almost all type of hydrolytic enzymes. So what does your lysosomes have, students? Lysosomes basically have the hydrolytic. They have the hydrolytic enzymes. They have hydrolytic enzymes such as your hydrolase, lipase, protease, carbohydrates. Hydrolase, lipase will break down lipids, protease will break down protein, carbohydrates will break down the carbohydrates. Optimally activate acidic pH. Now tell me, students, what will happen if I decrease the pH in lysozymes? Will the enzymes work or not? Simple question. Tell me, if you increase the pH from 4 to, if I increase the pH to around, let's say 10, pH increase to 10, will the lysosome work? Obviously not. Because lysozymes, for it to work, it should have an acidic pH. If you increase the pH, it becomes basic in nature and lysozymes cannot work that time. Cannot work. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acids. See here. This is what we learned here. This is the nucleus. Here we have ER, rough ER. Product is a protein. Packaging is done by Golgi body. This is the cis phase. This is the trans phase. Packaging is coming out. Product is here. One of the product is nothing but here. Lysosomes is one of the products of the Golgi complex. Packaging material. Okay, packaging material. Next concept is vacuoles. I believe there should be no doubt regarding that. That is function, how we get lysosomes. Lysosomes are also called as suicidal bags. Why? Because they completely break down things. Anything in the cell is not working. For example, inside the cell, mitochondria is getting destroyed. Mitochondria is not working properly. It will destroy the mitochondria completely. If the entire cell is dying, it will kill the cell only because it is unwanted now. It is not functioning properly. If anything is not functioning properly, there is no role. Kill it directly. Okay, that is what is happening in lysozymes. Now understand vacuoles. Understand vacuoles now. Vacuoles is very simple to understand. 1, 2, 3 main points are here. Understand that. First of all, remember, vacuoles are membrane-bound structures. Vacuoles are membrane-bound structures with a proper space. Membrane-bound space is there inside. Okay. Now, what do they store, students? Vacuoles is basically like the what do I say? Storage unit. It is a storage unit. Anything plant doesn't require or requires will be stored here. How you store your money in the bank, no? Or you have almirah in the house. In almirah or wardrobe, you keep all the clothes, all the requirement things. Even though some of that one dress you will never wear in your life, that pant or that shirt you will never wear in life, still you will keep it in that wardrobe. Same. Even in the case of vacuoles also, that anything which plants has, it will be stored in the vacuoles. What is stored here? For example, we can store here, plant can store water sap. Even the excretory products not equal to plants, store it in the vacuole. Okay. And students, remember, in the case of plant cell, vacuole is very large. The central vacuole is extremely large in the case of plant cell. Okay, it almost covers 90% of the cell volume. Cell volume is covered by 90% cell volume is covered by vacuole only. That big the vacuole is here. Very big vacuole. Because plant has a lot of waste. Plant has a lot of waste. Okay. Understand now. The vacuole is the membrane-bound space found in the cytoplasm. Membrane-bound space. It contains water sap, exudative products, and other minerals not useful to the plant. So dustbin now. It has made plants dustbin. Whatever is not required, it will throw there. Okay. The vacuole is bound by a single membrane called as tonoplast. Understand this. See, imagine this is the vacuole. This is the vacuole. The vacuole is covered by a separate layer called as tonoplast. Vacuole, a separate layer called as tonoplast. Very important. This is a cell. This is the vacuole. The layer of the vacuole is called as this is vacuole. Vacuole is covered by a layer called as tonoplast. Tonoplast. Okay, tonoplast. In plant cell, the vacuole can occupy up to 90% of the volume of the cell. That big vacuole is very large. Central vacuole is present. Okay. In plants, the tonoplast facilitates transport of number of ions and other material against the concentration gradient into the vacuole. Basically like active transport. Like students, remember always in your NEET score, if you're scoring anything around 700, you can easily drop to 500 if you don't study. But if you're scoring 500 and if you have to reach to 700, that is more difficult. This is happening against the gravity. So if you want to go from 500 to 700, don't you want to have to work really hard? So that's what is happening here. So here also, what are we doing here is, in plants, tonoplast is facilitating. In the case of plants, tonoplast is facilitating all from the low concentration to the high concentration. From low concentration to high concentration against the gradient, it is sending items inside. See? That's why inside the vacuole there is high concentration of ions. So against the gradient, from the inside, it is sending into high concentration. Yes, started only. Why you are shocked? You thought I won't start today? I won't do scam, don't worry. Okay, I'm not NTA. Now, hence their concentration is significantly higher in the vacuole than in the cytoplasm. What concentration? Ion concentration. Ion concentration is high in the vacuole compared to the outside. Now, in amoeba, contractile vacuole is an important for osmoregulation and excretion. See, if this is the amoeba, an amoeba will have something called as contractile vacuole. This contractile vacuole will make sure all the waste is removed outside. It also helps in maintaining the salt balance, osmoregulation. In many cells, as in protist, example is paramecium and all. Paramecium, food vacuoles are found by the engulfing the food particles. So there are food vacuoles in the case of protist. So vacuoles, let me revise everything. In the case of plants, vacuole is membrane bound, very large. Function is what? Function is dustbin. All the waste material is stored inside the vacuole. Okay. Vacuole has an outer layer called as tonoplast. Now, what is the function of vacuole? Vacuole can sometimes, what happens? It can transport ions into the vacuole against the gradient. Okay, against the gradient. Then we have the contractile vacuole in the case of amoeba, which help in osmoregulation, first point, help in excretion, second point for amoeba. Then there is also food vacuoles in the case of paramecium, that is your protist. Okay. Now we come to mitochondria. Any doubt here? Quickly ask me. Any doubt here, students? Quickly ask me. In the vacuole and Golgi complex. Any doubt? No doubt. Perfect. Now we'll go to mitochondria. Now tell me, what comes to your mind when you think of mitochondria? What comes to your mind? Or what that one line comes to your mind? No, one line comes to your mind. Mitochondria is called as what? Mitochondria is called as power house of the cell. All of you remember this dialogue. It's called as powerhouse of the cell. Yes, it is called as powerhouse of the cell. Why? Because mitochondria can produce ATP. ATP, that is the energy currency. It produces ATP. Okay, it can produce ATP. Now students, if you look at a mitochondria, how does it look like? How does it look like? The shape of this mitochondria. Doesn't it look like a sausage? Tell me. Doesn't it look like a sausage? Yes. Sausage shape is there. Yes. Sausage shape. What happens here? Site for aerobic respiration. The meaning of aerobic respiration is that respiration which requires the oxygen. Aerobic respiration happens here. Now, if you look at the diagram of your mitochondria, how many layers do you think there are? How many layers? Can you see here? Outer and inner membrane. So can I tell mitochondria is double membrane structure? It is double membrane structure, mitochondria. Yes, obviously, double membrane structure. Now let's see what is given here. Mitochondria, unless specifically stained, are not easily visible under the microscope. It means that they are so tiny and very invisible almost. The number of mitochondria per cell varies depending upon the physiological activity of the cell. For example, you tell me, do you need more mitochondria in your heart or do you need more mitochondria in your cheek cell? Tell me. Where do you need more mitochondria? You tell me, sir, more mitochondria is required in the heart cell because the requirement is more. So based on the requirement in heart cell or cheek cell, depending upon that, number of mitochondria will vary. So in heart cell there will be more mitochondria, cheek cell very less mitochondria because there is no requirement. There is no requirement. Okay. In terms of size and shape also, considerable degree of variation is also observed. Typically, it's a sausage shape. I told you, sausage shape, cylindrical, having diameter. They will not ask you this, of 0.1 to 1 micrometer. Length is 1 to 4.1 micrometer. Each mitochondria has a double membrane bound structure with outer membrane and inner membrane. Okay, students, let's draw the diagram once. If it's drawn here already, let's see. See here, there is an outer membrane, inner membrane. In the inner membrane, students, it has a foldings to increase surface area. Can you see? The foldings are called as cristae, and the matrix is called as matrix here. Obviously, the matrix is the matrix in which the Krebs cycle takes place. Matrix. Okay. Now, apart from this, there will also be DNA here. There will be double stranded DNA. Double stranded DNA will be there. Ribosomes, yes, there will be ribosomes here. There will be ribosomes also here. Okay. Ribosomes, which are 70S. Just like your chloroplast. Okay. Cristae, double stranded DNA. Double stranded DNA also here, okay, and cristae and everything. Now, apart from this, there will be certain racket-like structures here. There will be certain racket-like structures. This racket-like structure are called as elementary particle. Elementary particles also called as F0 F1 complex. F0 F1 complex will be present in the cristae. F0 F1 complex particle, also called as oxysomes. Or elementary particle or F0 F1 complex. It is found in the entire mitochondria. This is structure of F0 F1 complex. You see, this is F0 F1. F not an F1 complex. See, outer and inner membrane dividing in the lumen distinctly two equal aqueous compartments. Outer compartment and inner compartment. The inner compartment is filled with dense homogeneous substance called as matrix, where on the inner side, on the inside, now the outside membrane forms the continuous limiting boundary of the organelle. You see here, outer membrane is forming a continuous layer, outer membrane. Okay, the inner membrane forms a number of infoldings called as cristae. So inner membrane will have lot of foldings in a membrane towards the matrix. The cristae increases the surface area. The two membranes have their own specific enzyme association with the mitochondrial functions. Mitochondria are site for aerobic respiration, already discussed. They produce energy, cellular energy in the form of ATP, already discussed, and they are called as power of the cell. Okay. The matrix also possesses a single double-stranded DNA, 70S, a few RNA molecules, ribosome 70S, components required for synthesis of protein. The mitochondria can divide by fission. Last line is very important, PYQ. Mitochondria can divide by fission. Okay. Ads, unfortunately I cannot control the ads. Sorry. Ads will come and go, don't worry. Any doubt in this paragraph, quickly ask me. Any line you don't understand. Any line you don't understand, quickly ask me. Any line of NCERT you don't understand, or any of you? All of you know what is mitochondria. All of you know what is called the power of the cell. Two membrane, outer membrane, inner membrane. Yes? 70th ribosomes. Circular DNA. No doubt, right? This is always a habit of me, always asking if there is doubt. Okay. That's all. Now, let's do plastids. Again, very simple, students. Plastid is basically the which has the pigment structure, which has the pigments and all. Right, that is the plastid. Right, it is present everywhere. This particular plastid can provide in particular color to a plant. For example, chloroplast. Chloroplast will give you green color. Right, this chloroplast will give you green color. Similarly, leucoplast does not give any color but it has other functions. Then there is chromoplast, which imparts other color also. Okay, so let's understand one by one what exactly is the meaning of plastid and why this plastid is only present in the case of plant. Okay, why present is only in the plant. Plastids are found in all plants, cells, and in euglenoids. Again, it's a PYQ. In euglenoids. These are easily observed under the microscope as they are very large. So you can easily observe the plastid. Okay. They bear some specific pigments, thus imparting specific color to the plant. I told you, very specific pigments are present in the plastid. Based on the type of pigment, it can be classified into chloroplast, chromoplast, and leucoplast. Okay. Three different types. Is there a space to write? Yes, there is space to write here. Understand. One by one, I'll tell you. Okay, one by one, I'll tell you. Okay. First, we'll write chloroplast. Understand. Chloroplast, main function is trapping the sunlight. Why? For photosynthesis. Chloroplast. Chloroplast, there are two types of pigments there. One is chlorophyll. We have chlorophyll A and chlorophyll B. Other one is carotenoids. Under chloroplast. Then we have chromoplast. Chromoplast mainly has two things again. Where we have, okay, what happened? Where we have carotenes as well as xanthophyll. Both are present. Right? Carotenes and xanthophyll both are present in the chromoplast. They provide color. They give color. Which color? Yellow color, obviously. Yellow to orange and even red sometimes. That is chromoplast. Okay. Aditya, guessing what? Take my name. What is there in the name? Chill. Last one is leucoplast. Leucoplast is colorless. It is colorless. Leucoplast is completely colorless. Okay. In leucoplast, there are three types again. We have a myeloplast, elioplast, as well as aleuroplast. Amyloplast, amylose, amylopectin. Amylose and amylopectin are a type of starch, right? Starch. So it will completely store the starch. Or can I say carbohydrates? Carbohydrates. Now about elio. What about elioplast? Right? Elio, oil. Sounds like oil, right? Oil and fats. Aileron layer. It's a proteinaceous layer, right? So stores protein. It can store protein. So there are three different types of plastids: chloroplast, trap sunlight, photosynthesis; chromoplast, carotenes and xanthophyll, color; leucoplast, three types: amylo, elio, and eleuro. Amyloplast provides starch and carbohydrates. Elioplast, oils and fats. Aleuroplast provides protein. In examination, they can ask you directly any one of them. They can ask you, elioplast provides what? And mostly such questions will come in match the following. Always, such questions will come in match the following. Remember that. In match the following. Okay. Now, let's see what is given here. The chloroplast contains chlorophyll and carotenoids, pigments. Told you. Chloroplast has what? Chlorophyll and carotenoid pigments, which are responsible for trapping the light energy essential for photosynthesis. In chromoplast, fat-soluble carotenoids, pigments like carotenes, xanthophyll, and other pigments are present. Where? In the case of chromoplast. See, chromoplast has carotenes and xanthophyll. Color pigments will give you this color, following color. Okay, yellow, orange, red color. Leucoplasts are colorless plastids of variable shape and size. Amylo, carbohydrate, starch. Potato example. Elio, oil and fat. Eleuro stores protein. So directly whatever is given there, we have made it into points so you can understand. Now students, now we have something called as section of chloroplast. All of you know chloroplast. We've been studying about chloroplast and photosynthesis in higher plants. So many times we've studied. Students, here understand, your chloroplast is also double membrane. It is also double membrane in nature. Look here. We have the outer membrane and inner membrane. Both are present here. Then we have each one. You see each coin here. Each coin is called as thylakoid. The entire stack is called as grana. Each one is called as a thylakoid. Entire stack is called as grana. Okay. Now, between the two grana, can you see there's a connection? Can you see this connection? Between the two grana, the connection is called as stroma lamellae. And inside the chloroplast, there is a matrix. The matrix is called as stroma. Is this recorded? No, it is not recorded. Fareen, it is not recorded. You can ask your doubts. I will clear your doubts also. You can shoot your doubts anytime. Okay, so three different types of chloroplast. Sorry, three different types of plastids: chromoplast, chloroplast, and leucoplast. Okay, directly question can come from any part of this. Any part of this question can come. Okay, so any doubt you quickly ask me. Shoot. Any question here? In plastid. Now here students, I'll tell you some extra information, understand, extra information, okay. Remember, in this stroma lamellae and thylakoid, in both the places we have the light reaction happening. Light reaction is happening here, okay, light reaction is happening. In the stroma right here, dark reaction takes place. Dark reaction, remember that, okay. On this, now, majority of the chloroplast of green plants are found in the mesophyll cell of the leaf. Okay, these are lens-shaped, oval, spherical, or discoid. Okay, their number varies from 1 per cell. That is, chloroplast can vary from only 1. In Chlamydomonas, there is only 1 chloroplast, and it can vary up to 20 to 40 per cell in the case of mesophyll cell. So, in the case of mesophyll cell, it will be present in higher number. Okay. Like mitochondrial, chloroplast has also double membrane bound. Of the two, inner chloroplast membrane is relatively less permeable. So inner membrane is less permeable compared to the outer membrane. Okay. Now, a space is lined by the inner membrane of the chloroplast is called as stroma. That is the matrix. A number of organized flat disc-like structure or the coin-like structure are called as thylakoid. The entire stack is called as grana, are present in the stroma. The thylakoids are arranged in a stack like pile called as grana or granum or integral granule thylakoids. In addition, there are flat filamentous tube like called as stroma lamellae and stroma lamellae students. The stroma lamellae helps in the connecting the two different stack of coins. Okay, so from one grana to second grana, the connection is between the stroma lamellae. Okay, connecting the thylakoids of different grana. The membranes of thylakoid enclose a space called as lumen. Now, where is the lumen here? Understand. This is the thylakoids, right? These are the thylakoids. And this, between the two thylakoids, we have the connection, stroma lamellae. Okay. If you zoom into one of the thylakoid, you will end up getting like this, something like this. This is the membrane. The middle part, can you see? This part is called as the lumen. This part is right here is called the lumen. Lumen of thylakoid. The middle part of the thylakoid. Okay. The stroma of chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. What does it mean? It means that stroma has all the enzymes required for photosynthesis. Quickly give me an example, any of you. Quickly give me an example of an enzyme present in the dark reaction or Kelvin cycle. Dark reaction and Kelvin cycle, which enzyme is present? Quickly give me an example. One example. Rubisco. Yes, Rubisco is the enzyme. Rubulose 1,5 bisphosphate. Rubisco oxygenase carboxylase. Right. So Rubisco enzyme is present in the stroma. Right. Now what about in the case of C4 plant? C4 plant, in the mesophyll cell there is one more enzyme, no? In mesophyll there is one more enzyme in C4 plants. In the case of C4 plants, they have something called as pepcase. Pepcase is also present. Remember those points. Plant physiology. Okay. Now it also contains small double-strand circular DNA also present. So it shows you that chloroplast and mitochondria, they have their own DNA. They also have their own ribosomes. And both the ribosomes in the case of your chloroplast and mitochondria are 70S ribosomes. 70S ribosomes. Both. 70S ribosomes in the case of both chloroplast and mitochondria. Okay. Chloroplast pigments are present in thylakoid. Ribosomes are present. See? Is present. Now we'll talk about ribosomes now. Ready? Ready, students? Any doubt in chloroplast? Every single line is done. No doubt? Now we'll do ribosomes. Okay. Ribosomes are granular structures first observed under the electron microscope as a dense particles by George Paldane. So George Paldane was this very important PYQ again. George Paldane was the scientist who observed the ribosomes for the very first time in the year 1953. They are composed of ribonucleic acid and protein. So if someone asks you, students, what is ribosomes made up of? What is ribosomes made up of? They are mainly made up of RNA and protein. RNA and protein. Okay, RNA and protein. Okay. And they are not surrounded by any membrane. So remember, ribosomes is found in both prokaryotes and eukaryotes. In both prokaryote and eukaryote, ribosomes is without a membrane. It is always without a membrane. The eukaryotic ribosome are ATS. In the eukaryotes, it is ATS. While prokaryotic ribosomes are 70S. So prokaryotic 70S, eukaryotic 80S ribosome. Each ribosome has two distinct subunits, a large and a small subunit. Okay, for example, this is a ribosome. Ribosome will always have two units, something like this. This is the smaller one. This is the larger one. Two different subunits. Okay. No. The two subunits of ATS ribosome includes 40 and 60. So ATS ribosome has what? 40S plus 60S. Now you might be wondering, sir, 40 plus 60 is 100. How can you tell 80? Because students, this is not additive. It is 40 and 60 are the values derived from the where the on the sedimentation coefficient. For example, if you can notice here, if you take this ribosomes and add in a test tube, if you take the ribosomes and if you add it into a test tube like this, if you add into test tube, in the bottom we will have the 100. We will have the ATS ribosome. We'll have the ATS ribosome. ATS. When you add ATS inside completely, once you split it open, okay, small subunit and large subunit. Here you will find 60S. Here you will find 40S. Basically, how a ribosome will dissolve and what level it will, sediment. That is called a sedimentation coefficient, and based on the sedimentation coefficient, we give the number as 30-50 or 40-60. Not additive thing. It is not additive. Okay, it is not additive in nature. See here. 80S ribosomes are 60S and 40S, while that 70S ribosome. Friends, remember, the 70S ribosome is mainly found in the prokaryotes. Prokaryotes, mitochondria, and chloroplast, as well as chloroplast. Everywhere you find the same enzyme. Okay. Here S means what? Sedberg unit. S means Sedberg unit. Sedberg unit. S means Sedberg unit. Stands for sedimentation coefficient. I told you how well a particular subunit or the entire unit can sediment. Based on that, we have the value, and value is called as the S value, sedimentation coefficient value. Okay, sedimentation coefficient value. Okay. Now, it is indirectly a measure of density and size. Both 70 and 80S ribosomes are composed of two subunits. Can you see here? In the case of prokaryotes, we have a small subunit is 30S, 50S, we get 70S totally. In the case of eukaryotes, we have 40S, 60S, totally is 80S, not 100. It is 80S. Okay. Good evening, Angel. Okay. It is 80S. Now, students, with that, ribosome concept is also over now. Ribosome concept is also over. Now. Can we start with cytoskeleton now? Should we? शुरू करें cytoskeleton. Students, let me tell you, cytoskeleton. Okay, it's very simple to understand, but many people get confused here. Understand, like how our body has a bones as a framework. How our body has framework, even a cell has a framework to support. Like how our body has bones to support our body, ourself, everything. Plants also have certain skeleton. The skeleton in the case of plants is called as, not plant, sorry, in the case of cell is known as cytoskeleton. So it is basically the network of, filamentous proteinaceous structure. How our body is supported by bones, a cell is also supported by the cytoskeleton. Okay, cytoskeleton. An elaborate network of filamentous proteinaceous structure consisting of the microtubules, microfilaments and intermediates. Students, understand, there are 3 types of cytoskeleton now. 3 types. First one is called as microtubules. Second one is called as microfilaments. The third one is called as intermediate filament. Now, what is the difference between 3? In microtubules, it is completely hollow. Microfilaments are solid, and intermediate filaments are also hollow in shape, Your microtubules are made up of tubulin protein, while your microfilaments are made up of actin and myosin. They are made up of certain non-contractile protein. Now, compared to microfilament and microtubules, microfilaments are a little bit flexible. Like how muscle contraction. In the muscle contraction, you might have heard about it. Actin and myosin. So they can contract easily. But here, they are not able to contract. Non-contractile. Microtubules cannot contract or relax. Now you will ask me, sir, if microtubules are not able to contract and relax, what is the main function of microtubules then? Understand, students, if they are not able to contract or relax, their main function of microtubules is assembly. I'll show you a simple diagram, you'll get a super idea. See this. This right here is a microtubule. Can you see? Each one is hollow. Each one right here is hollow. Right, they're hollow. They are called as microtubules. What they do is they arrange themselves and they're functional later on. They arrange themselves. I'll show you one more diagram. Microtubules. Can you see? Here. All the microtubules which are hollow are arranged like this. They make up the skeleton of a cell. They're arranged. You can see they're arranged properly. Cytoskeleton. Okay. That's what is given here. So where do you find microtubules? Microtubules are found in cilia, flagella and centrioles. So centrioles, flagella, cilia, everything is made up of microtubule. It is made up of microtubule. Okay. Filaments. Cytoplasm in collective referred to as cytoskeleton. All 3 together are called as cytoskeleton. The cytoskeleton is in a cell are involved in many functions such as mechanical support, motility, which is a PYQ, maintenance of shape of the cell. All these are functions of cytoskeleton. First one is mechanical support, obviously framework. Motility, it helps in movement. Maintains the shape of the cell, like how bones maintain the shape of our body. Maintain the cell, maintaining the shape of the cell. Okay. Now we come to cilia and flagella. Okay. What is cilia and flagella, students? What is cilia and flagella? First of all of you know, cilia and flagella basically like hair-like outgrowth on the cell wall, cell membrane. Hair-like outgrowth on the cell membrane. Let's see what is given here first of all. Then we can come to the diagram. Okay. Cilia or cilium or flagellum are hair-like outgrowth has the cell membrane. Okay, of the cell membrane. See, or the present of the cell membrane. Cilia are smaller in structure. So write down difference. Cilia and flagella. Cilia is much more smaller and it shows oars-like movement. Oar-like movement. Actually, what is oar-like movement in next 2 minutes? Flagella is longer, helps in cell motility, cell movement, yes. And flagella is different in prokaryotes. Different in prokaryotes. Okay, let's read what is given here. Cilia has small structure which look like a or causing movement of either cell or surrounding fluid. See, this is or, student. This is or. This is called as an or. You might have seen, right? So when you're pushing it, can I tell, are you going forward? Yes, you're going forward. When you're trying to push it like this, can I tell water is being pushed back? Yes. That is called as or-like movement. So it's given here. It works like an or, causing the movement of either the cell, either the board will go front, or the cell will go front, or the surrounding fluid will go back. See, or the fluid goes back. Flagella are comparatively longer and responsible for the cell movement. Done. The prokaryotic cells also possess flagella, but these are structurally and different from that of the eukaryotic flagella. So prokaryotic flagella, eukaryotic flagella, both are different. Very different actually. The electron microscope study of the cilia flagellum shows that they are covered with a plasma membrane. Their core is called as axoneme. Understand? Let's do the structure of the flagella, cilia and flagella. Cilia and flagella. Okay, they have an outer layer called as the plasma membrane. Inside the plasma membrane, they have something called as Ganji chashma. Okay, it looks like a chashma, but it's not. It is basically the middle part is called as your. I'll tell you. Let me draw it first. Three, 4, 5, 6, 7, 8, 9. Okay, there are 9 of them. One, 2, 3, 4, 5, 6, 7, 8, 9. Okay. Now, students, understand. In the structure of the cilia and flagella, I told you, cilia and flagella is made up of what? Microtubules. Since they cannot contract and relax, they have to form a group and gang and everything. So this is the gang here. Now here, if you can notice, this outer part, the red part, they are called as peripheral microtubule. What about the center part? This is called as center microtubule. Now, what is this then? This rod-like structure which is connecting the central to periphery. The rod-like structure is called as radial spoke. Radial spoke. Now, this is periphery, this is central. Can you see this one line, one more here? This dark color green one, where the radial spoke are attached. This dark color green one is called as what? That is called a central sheath. Central sheath. Central sheath. Okay. Now, how do you think the two different microtubules talk to each other? Can you see this one? This is called as linker. How? Peripheral microtubules are attached to each other. Peripheral microtubules are attached to each other with the help of linkers. These are called as linkers. Can you see? Linker. Linker, periphery microtubule, radial spoke, central microtubule, central sheath. That's all the structure of cilia and flagella. That's all the structure of cilia and flagella. Okay, do you understand? It is made up of microtubules, peripheral microtubules, central microtubule. Central microtubule are attached to the central sheath. Central sheath is attached to the peripheral microtubule with the help of radial spoke. The peripheral microtubules are attached to each other with the help of linker. B to A linker. B to A linkers. You see, B to A linker is there. That's all. Now, student, this entire core part, can you see? Where did the diagram go now? This entire core is called as axoneme. Entire core is called as the axoneme. If you can compare here, look, let's look at here. See, we have the plasma membrane, peripheral microtubules which are present as doublets. I've told you doublets. Present as doublets on periphery, doublets. Doublets. See, present as doublets. Index which is basically the linker. Central microtubule. Radial spoke, how they are attached to central sheath. Central sheath. This is called as central sheath. Each and every point covered. This is the electron microscope image. Electron microscopic image. Okay. So, core is called as axoneme. They possess a number of microtubules running parallel to the long axis. Axoneme usually has 9 doublets of radially arranged peripheral microtubules. See, 9 periphery. So, you tell me, there are 9 in the periphery microtubules. In the center, there are 2 microtubules. So, can I call it 9 plus 2 arrangement now? 9 plus 2 arrangement? Yes, I can call it 9 plus 2 arrangement. Okay. A pair of centrally located microtubules. Such an arrangement is of axonemal microtubules is referred to as 9 plus 2 array. The chemical tubules are connected by a bridge and is also enclosed by a central sheath, discussed already, which is connected to the one of the tubulas of each peripheral doublet by radial spoke. So we have radial spoke here. You see, radial spoke is coming out. It's coming out. Thus, there are 9 radial spokes. Peripheral doublets are also connected by a linker. See, linker. Both cilia and flagella emerge from the centriole-like structure called as the basal body. See, last line you understand. Both cilia and flagella emerge from the basal body, right? See here. See, this is called as the basal body. From the basal body only cilia and flagella are coming out. Cilia and flagella is coming out from the basal body. See here. This is the basal body. All the microtubules solid. They are solid. They're coming out. Here. Flagella and microtubules are out here. Now we will do centrosome. Now we will do centrosome and centriole. Can we start? After centrosome, we have only few left. Centrosome is there. Then we have nucleus, and we are done naturally. This chapter is over after that. Now we will do centrosome and centriole. Centrosome and centriole. Understand, students? I think I told you before, when I was teaching you about non-membrane bound organelles, non-membrane bound organelles, I told you ribosomes and centrosome. Ribosome is present in both plants and animals. Plants and animals, but centrosome is only found in animals. Only in animals. Found only in the case of animals, centrosome. Now, if you look at the structure of centrosome, students, centrosome is made up of two centrioles, which are right angle to each other. One centrosome is made up of two centrioles. Can you see? Which are perpendicular to each other. Which are perpendicular to each other. Now, if you this centriole, students, this centrioles or this entire thing called centrosome has a amorphous outer structure. It has something called as a amorphous structure on the outside. You see here, amorphous structure on the outside. They call it amorphous pericentriolar material. Pericentriolar, or we can also call it kinoplasm. Kinoplasm. The outer layer, can you see this outer layer, dot layer? The outer layer is called as amorphous pericentriolar or kinoplasm, or we can also call it amorphous structure. Okay, we can also call it centrosphere also actually. Centrosphere, kinoplasm, amorphous pericentriolar material or space. Okay, this is how the centrioles look like. Now, students, if you cut the centriole, see here. If see one centriole, don't they look like cartwheel? They look like cartwheel. Can you see? They look like cartwheel. Each centriole. If you look at the centriolar structure, of centriolar, they are also made up of microtubules. Look at the structure of them. Look, structure is very important again. Understand. Here in the middle there is no. In the middle there is no this thing inside. In the middle there is no microtubule. We have a proteinaceous hub here. It's made of protein. Okay. Apart from that, there is 9 different radial spokes coming out. 9 different radial spokes are coming out. Okay. On the end of each radial spoke, there is a peripheral microtubule. 1, 2, 3, 1, 2, 3. So completely 9 radial spokes, each one with a triplet of microtubules. So again, this is called as triplet of peripheral microtubule. peripheral microtubule. This one. This is called as radial spoke again. Radial spoke. This is the peripheral microtubule done. Here we have B. We have one more, no, C. C, B, A, C, B, A, C, B, A. In all three cases, that is, peripheral microtubule is interconnected. Peripheral microtubule is interconnected to each other with the help of linker again. C2A linker. C2A linker. C2A linker in the case of centriole. This structure right here is one centriole, student. There are two centrioles. Out of the two centrioles, if you look one from the top also, you will see something like this. See here. It has peripheral microtubules, 1, 2, 3 triplets, proteinous hub, and linkers, and triplet of microtubule triplets are present. Okay. So with that being said, we only have nucleus to go, that's all. Once you finish nucleus, the chapter is over. Okay. So let's take a 20 minutes break and come back, and then we'll start again. At 7:25 we'll resume. Resume at 7:25. Okay. I'll go drink some water and come. I think So let's finish the last part of the class. It's very easy. And the nucleus, right? Let me draw it for all of you, and you will understand without any mistake. Okay, nucleus, the last part of today's class. Okay, nucleus. Now understand the diagram first of all. Understand the diagram of nucleus. I've always told you nucleus is a double membrane structure. Nucleus is a double membrane structure. This is the inner membrane, and we have an outer membrane. Can you see, students? We have two different membranes here, inner membrane and outer membrane. Okay. Now, this is the inner membrane and outer membrane. Let me just write for you. This is the outer membrane, and this is the inner membrane. Okay, inner membrane. Now what about the space between, students? This space here, this one is called as perinuclear space. Perinuclear space. Now here inside they should also have a matrix here inside. Yes, the matrix. The matrix is called as nucleoplasm. Inside is called as nucleoplasm. Right? No, this was evening snack. I went. Lunch I ate after the first class, right? Lunch was done after the biomolecules class. I ate the lunch, and now I ate the evening food. So I take 5 meals in one day. First breakfast, second breakfast, lunch, post lunch and dinner. 5 meals in one day. Right. Now students, you need to understand here is, in this perinuclear space, there are small holes. Obviously, from the nucleus, students, from the nucleus, your DNA, your RNA has to come out. Some form of RNA proteins. RNA proteins has to travel. There are small gaps here. Okay, there's some small gaps in the perinuclear space. Will happen when the outer membrane and inner membrane, when both of them will join together, right? When outer membrane and inner membrane join together, they make up small gaps. These small gaps are called as nuclear pore. Nuclear pores are here. Nuclear pores are present. Okay. Now here you need to understand here is, once nuclear pores are here, I have always told you from the outside, the endoplasmic reticulum will always be continuous. From the outer wall, endoplasmic reticulum will always be continuous from the outer wall. Now what about the inside? What do we have in the inside? Do we have the genetic material? Yes, we have the genetic material on the inside. We have long threads inside. Genetic material is in the form of threads. These threads which are here are called as chromatin threads. Why? Because they are in the interface right now. Interface. Thread-like structure in interface. Now, apart from this, there is also something called as ribosome factory. A nice factory is also inside. If you notice, there is one factory on the inside called as ribosome factory. On the inside, there are a lot of free-floating ribosome factories. These inside ribosome factories are called as, students, can you see? These ribosome factories are called as nucleolus. Also present on the inside. Nucleolus is also present on the inside. It's called as ribosome factory. Now let's read what is given here. Nucleus as per cell organelle first described by Robert Hooke. Right, dead cell discovered by the Robert Brown. Right, so Robert Brown. Robert Hooke is dead cell. Robert Brown is nucleus. Robert Brown. Okay. As early as 1831, later maternal of the nucleus stained by the basic dye was given the name chromatin by Fleming. So chromatin was found by Fleming. Okay. The interface nucleus of the cells which is not dividing. Can you see? This is called as interface nucleus. Interface nucleus right here. Okay. Has highly extended and elaborate nucleoprotein fibers called as chromatin on the inside. Nuclear matrix called as nucleoplasm. And one or more spherical body called as nucleoli or nucleolus. It's not written properly. I think you can't see this. It's called as nucleolus. nucleolus. That is a ribosome factory. Okay, ribosome factory, nucleolus. Okay. Electron microscopy has revealed that the nuclear envelope, right, the outer wall, which consists of two parallel membranes, see, outer membrane, inner membrane. Between part is called as perinuclear space. Perinuclear space is the between them. Forms a barrier between the material present inside the nucleus and that of the cytoplasm. Inside the cytoplasm. Okay. Now, the outer membrane usually remains continuous with the endoplasmic reticulum here. ER. It is continuous with the ER. Yes, continuous with the ER. And also bears ribosomes on it. So you can find ribosomes on it. If it means it is RER. If there is ribosomes, it is called as rough endoplasmic reticulum. Obviously, right? Rough endoplasmic reticulum. No. Bears ribosomes on it. A number of places at nuclear envelope is interrupted by minute pores. The minute pores are called as what? Nuclear pores. Here. You see here? Minute pores are called as what? Nuclear pores are present, also present. Okay. Which are formed by the fusion of two membranes. I told you, when the two membrane fuses, the outer membrane, inner membrane, when both of them fuse together, they make the nuclear pore. Okay. These nuclear pore are important for movement of RNA and protein molecules inside and outside of the nucleus. Okay. Now, normally there is only one nucleus per cell, only one. But sometimes what happens is a particular cell can have multiple nucleuses, right? Variation in the number of nuclei can also frequently be observed. Okay. Some mature cells even lack nucleus. Example, the famous example is the red blood cell. Your red blood cells do not have any nucleus, they are anucleated condition. And that is one of the main reasons, red blood cells, in them, only and only glycolysis takes place. There is no Krebs cycle, there is no electron transport chain. Okay. Now, the nuclear matrix is called as nucleoplasm. I told you, inside part is called as nucleoplasm containing nucleolus. It contains nucleolus. What is nucleolus? Ribosome factory. It creates ribosome and also creates rRNA. Okay. See, I told you. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm. It is not membrane-bound structure. It will be free-floating for rRNA synthesis. rRNA synthesis. Ribosomal rRNA. Factory for rRNA synthesis. rRNA synthesis happens here. Where nucleolus. A large or more number of nuclei are present in the cells actively carrying out proteins. Why more? Because if plant require or animal require more proteins, it means they require more RNA. If they require more RNA, it means that they require more nucleolus. They require more nucleolus. So more nucleolus, when is it required? When more protein is required. That's all. No. Next is the diagram of your chromosomes. Very important, very easy to understand. See, a particular chromosome has something called as a primary constriction. The primary constriction is called as centromere. Okay. Now on this primary constriction, there are two sister chromatids. They are the sister chromatids. Okay. Everything got erased. Oops. Gone. Let me draw it again. We have the primary constriction called a centromere. Right. Now on this primary centromere, right, we have the sister chromatids. We have the sister chromatids. Now on the sister chromatids, students, the end part, can you see this? This end part is called as telomere. Now on this primary sister chromatid, there is also a secondary constriction. Can you see? This is called as secondary constriction. Secondary constriction is transcriptionally active, and the secondary constriction is called as satellite. It is called as satellite. Satellite. Okay, satellite. And in your, out of 23 pair of chromosomes, only 5 pair has a satellite. That is basically the 2, 21, 22, 13, 14 and 15. Only these 5 of them has the satellite, the secondary constriction. Okay. Now around the primary constriction there is some form of a protein layer. Protein is disc-like structure around the centromere. Around the centromere, this protein layer is called as kinetochore. It's called as kinetochore. Now, what is the function of kinetochore, students? The main function of kinetochore is that during the spindle fiber attachment, during the cell division, when the centromere is to split open, or when the homologous chromosomes are separate, kinetochore is the point of attachment of the spindle fibers. Can you see? Spindle fibers are attaching to the kinetochore. They do not attach to centromere. All the spindle fibers attached only and only to the kinetochore, not to the spindle fibers. Not to the spindle fibers. Okay. Done. Let's see what is given in NCERT now. You may recall that interface nucleus has loose and distinct network of nucleoprotein fibers called as chromatin. There is chromatin. Okay. But during different stages of cell division, metaphase, anaphase, prophase, right, showed structural chromosomes in place of nucleus. So in the nucleus we have chromosomes. All of you remember, right? PMAT. Prophase, metaphase, anaphase, telophase. In these phases we can see the nucleus. We can see the chromosomes. Right. Chromatin contains DNA, some basic proteins called as histone, some non-histone proteins, and also RNA present inside the nucleus. A single human cell has approximately 2 meter long thread of DNA. 2 meter long DNA inside one cell. 2 meter long. And they said that if you remove all the DNA from a particular human, you can make a ladder to the moon. That much of DNA is present in your body. Appreciate that. Right, appreciate that. You will study the details of the DNA packaging in the class 12 molecular basis of inheritance. Okay. Every chromosome visible only during the dividing the cell essentially has a primary constriction called a centromere. Here. Primary constriction, centromere. On the sides of which disc-shaped structure called as kinetochore. Kinetochore is also present. Kinetochore. Okay, kinetochore. Centromere holds the 2 chromatids of chromosome, sister chromatids. Based on the position of the centromere, chromosomes can be classified now. See, for example, it can be classified into metacentric, submetacentric, telocentric, acrocentric and telocentric. See here. You can see here. It is somewhere here. See. V L J I. V L J I. V is in the case of metacentric, submetacentric is L, acrocentric is going to be J shape, and telocentric is going to be I shape. Based on the location of centromere, if it's completely terminal, almost terminal, in the middle, or away from the center, in which one is longer, other one is going to be little shorter. Depending upon the position of centromere, we can have different types of shapes of chromosome. VLJI. VLJI. Okay. This is a very famous question, let me tell you. Very famous question. Okay. Now, the metacentric chromosome has middle centromere forming 2 equal arms. Can you see here? In metacentric, can you see 2 equal arms? That is P arm and Q arm. Are they equal? Obviously they're equal. PU is equal to Q here. In the case of metacentric, here also you can see P arm and Q arm both are equal. So V shape. Okay. Now, the submetacentric chromosome has centromere slightly away from the middle. Can you see? In submetacentric, it is slightly away now. Here it was in center, dead center. Now it is little away from center. Little away from the center here. This one is shorter, this one is longer. P arm is longer, Q arm is little shorter here. Yes, Q arm is little shorter here. That is submetacentric. Okay. Now, resulting in one shorter and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming the extremely short and one long arm. In acrocentric, it is almost towards the terminal. Can you see? Almost towards the terminal. One is completely short arm, one is completely long arm here. See, you can see one is completely short, one is completely long here. That is an acrocentric, which gives the distinct J shape. J shape. J shape. Okay. Now, last one. Understand. Telocentric chromosome has a terminal centromere. No. One arm is completely missing in telocentric. In telocentric chromosome, can you see here? Extremely terminal. The two arms are almost missing here. Can you see? One more arm is only missing here. So we have telocentric. Telocentric is I shape. Telocentric is I shape. So this is actually one chromatid. One chromatid is represented everywhere. One chromatid is represented everywhere. Based on the position of centromere, we can have different types of chromatid. Like, metacentric is V, submetacentric is going to be L, acrocentric is going to be J, and telocentric is going to be I shape. It's going to be I shape. VLJI. VLJI. Sometimes a few chromosomes have non-staining secondary constriction. Told you. Secondary constriction. That is called as satellite. Satellite on the top. Okay. Called as satellite. At a constant location, this gives appearance of a small fragment called a satellite. Last concept is called as microbodies. Very easy to understand. Nothing. Many membrane-bound minute vessels called as microbodies that contain various enzymes are present in both plants and animals. Small vesicles which has their enzymes. Micro means small bodies, small bodies of small structures with a lot of enzymes in it, found in both plants and animals. That's the end of the class, students. That's how you do the end. That's the end of the class. Now, students, if you have any doubt, right? Any doubt regarding any part, structure of centriole, see, structure of your centriole, structure of your flagella, structure of flagella. Or it could be, what is the difference between different type of ribosomes? What is the plastid pigment? Structure of mitochondria. Structure of your Golgi body. Endoplasmic reticulum in previous class I did. We did something called a cell wall formation. Can you see? Plasmodesmata. Structure of plasma membrane. What is flip-flop movement? What is lateral movement? What is bilayer? Okay. Now, we also did the plants and animal cell. We did something called as eukaryotic cell, where we did the different structures of envelope. Outer part is called as glycocalyx. Middle layer is called as cell wall. Inner layer is called as plasma membrane. So we have done the entire chapter of every single line of NCERT. Cell theory also we did. Three Scheiden, Swan and Rudolph-Virchow. 3 different people there. Students, with that, the chapter is over now. Now it's your duty to read the chapter again on your own. Yes, it is your duty right now to read the chapter again one more time. And then, if you have any doubt, you can let me know in the comment section. And or you can also always text me on my Telegram, where I always reply, obviously. Telegram is none other than Baswara Sir Biology. Baswara Sir Biology. Right. Always Telegram, you can always text me and you can ask me your doubts. Or if you want any particular notes, right, I will send it to you. Okay. So thank you so much, students, and have a great day ahead, all of you. Thank you so much, students.